Today we are going to discuss about a very important technique, how we can observe stomata which is present on the surface of the leaf in simplest way. That is why it is called as simple science. So these are various leaf samples with us. From there we will show you how in simplest way we can observe stomata, guard cells in effective way. So this is Syngonium leaf, first leaf. The second leaf is Thunbergia leaf, and likewise some other leaf samples are also there. So we have to take a leaf, fresh leaf must be there, and then we have to separate peel from the leaf. Peel from the leaf. How to separate peel? Peeling means removal of, this is called as peel, removal of lower epidermis. So this we have removed, lower epidermis has been removed by use of one very fine forceps, and this peel, that is called as peel. Now this white portion, this we have to put in one stain we have to use here, and the stain is we are going to use today toluidine blue only, right? Even safranin can be used, but toluidine blue is giving somewhat better kind of results, better clarity we will get. So here this is now in water only. We have to separate the peel from the remaining part of leaf. So stain is going to be added here. This is the toluidine blue, just a few drops of toluidine blue solution. Already ready-made solution is available in market, and little bit diluted. Not more concentrated, not more dilute also, so based on our requirement only. So the leaf sample with the peel also. Peel means the, here in our case it is lower epidermis. We have to give treatment for few. So after around 2 minutes of treatment of the toluidine stain, we have to take the sample in distilled water. This is plain distilled water only, that is also called as deionized water. We have to separate the stained piece, that is peel, from here and put on a slide. A slide we can mount this piece or the specimen or peel even in water also, because we are making science simple. No need of more complicated chemicals and all. Otherwise, as per scientific way, it must be in glycerin only. So in water drop we are separating it. So only the peel part, that is now blue in color because of the stained toluidine blue, we have to separate. Remaining leaf part is not useful for us because we are going to see the stomata only, and stomata generally concentrated on lower surface of the leaf only. So finally we have peel only, lower epidermis only. Remaining parts cut and discarded. With help of forceps and needle. So this is one glass slide only. Our peel sample is there. So this we have to cover with the cover slip. This we have to make in such a way that bubbles should not form. So slowly with help of a needle we have to slide the cover slip over the sample. Now this is covered with the cover slip. Now our slide is ready to visualize under the microscope. So excess water can be absorbed by use of tissue paper or filter paper. It will give a complete stability of the slide, cover slip and sample over the slide. So our objective is to observe stomata on the lower surface, lower peel of a leaf. So now slide is ready to be visualized under the microscope, and this is our same Leica microscope, one of the best available microscope, especially in our laboratory. So this is fixing of slide, and now switched on. Light is coming from the source of light, and now we have to adjust, focus and adjust. Our slide is there. Right. So again we have simplified this technology, that is why simple science. We are not going to use that camera that is attached with the mounted on the microscope. How? Because now everybody is having a cell phone with them. How a cell phone camera can be used to visualize the tiny cell phone camera we are using to visualize the tiny stomata which are present on the peel, that is lower epidermis of the leaf sample we have taken today, just now. So this is at low power. This is at low power, right? These blue structures you can see here, these are all stomata only, right? We will put on high power and then we will see the wonder how stomata appears. We have changed now the power. You can see here it is 40x, right? Objective piece is objective lens is 40x. So after that, how the stomata, same stomata appears in the cell phone camera. These are these blue structures you can see, these are stomata. Pores you can see, stomatal pore. Yeah, very clear. No much technology is needed. Cell phone, now everybody is having cell phone in hand. Right. These two cells you can see around the pore, these are called as guard cells. These are called as guard cells, two guard cells. Then around the guard cells, center is the pore of the stomata, and around the pore these are two guard cells. Around the guard cells, these 2 or 3 cells are there here. These are called as subsidiary cells, and around the subsidiary cells these are folded red structures you can observe. These are called as epidermal cells. So on lower epidermis of a plant, how stomata are present? These are almost half open stomata, because now the time is around 1 o'clock. This is the time when stomata are going to close again in nature, because light intensity, sunlight intensity is getting more and more, right? And if we collect the sample, leaf sample in morning, we can observe. So we have to collect in time. If we collect the samples in morning, early morning around 9 o'clock, stomata remain fully open. Pore used to be a big in size. So these are all stomata, which we cannot imagine to see through naked eyes. Even simple microscope also we can observe, but this much of clarity with simple instruments, because now everybody is having cell phone, right? No need of a mounted camera on the microscope also. See, this is the clarity of even you can see here inside the guard cells you can see these green structures. These are chlorophylls present in the guard cells of stomata. So this is the simplest way of observing stomata, tiny pores, by use of simple technology, by use of microscope and by use of cell phone camera. We can make videos, we can make photographs, right? So with different powers, objective lens powers, we can increase their resolution, we can increase their magnification, these things, and we can observe. So hope you understand the simplicity of science, the wonder of science,